So first of all, thank you for this opportunity to present this. I will be talking to you about basically the topic of my PhD thesis, so glycosylation in Down syndrome. And first of all, usually I like to start my presentations by defining the things from the title, and I will gladly skip over glycosylation this time. And I will just tell you briefly something about Down syndrome. So I'm pretty sure that we all know that Down syndrome is actually a genetic condition that is caused by either partial or full trisomy 21. The thing about Down syndrome is that its clinical picture is actually very variable. And these people who have Down syndrome, they often show a lot of different, comorbidities. It's called a syndrome because it encapsulates a lot of different symptoms, right? So I won't be going into the details and intrinsics of Down syndrome and all of that. But I just want to really briefly mention some of the things that are actually quite related to what I will be talking about later. So first of all, if you look at Down syndrome already on the level of the cell, you can actually see, so when you take primary cells from a person with Down syndrome, it actually shows a lot more DNA damage than cells from people who are the same age. But they don't have Down syndrome. So already on the level of the cell, you can tell that the cells are going into senescence or cellular aging earlier in Down syndrome. But then when you focus on the entire organism, you can see that these people actually develop really early onset dementia. So sometimes even before the age of 30, people are already diagnosed with Alzheimer's dementia, which is really, really early onset, basically. And another thing is they tend to develop a lot of different conditions that can all be grouped into this sort of category that we named aging-related reduction in tissue regenerative capacity, which would mean that they basically develop things like alopecia or osteoporosis or delayed wound healing and so on. All of these things, basically, I think I've mentioned the word aging like 10 times in the previous minute or so. So I think you can tell that what I mean to say is that Down syndrome is basically kind of a progeroid syndrome because it showcases so many signs of accelerated aging. And why that is interesting to us, well, as my colleagues have very nicely spoken before, we do a lot of immunoglobulin G or IgG glycosylation analysis in Genos. And the changes of IgG glycosylation have actually been correlated with many different physiological states and diseases, as you have already heard. But also, another thing that we have established is that IgG glycans are actually really good biomarkers of age. Basically what happens with age, there's more stuff that happens, but maybe the most pronounced change is this drop in G2, which means glycans that contain 2 galactoses. And you get an increase in G0, which are glycans that contain no galactoses. So that is basically something to keep in mind just for the rest of my results that I will be telling you about. And then we decided to combine those two things. So basically you have Down syndrome as a progeroid kind of syndrome, and you have IgG glycans that are markers of aging. So we actually have this published as a preprint, and hopefully it's going to be published very soon as an actual paper. Here it's dubbed, it's kind of a mouthful, accelerated biological aging in people with Down syndrome with full and segmental trisomy 21 begins in childhood as revealed by immunoglobulin G glycosylation. So what were kind of our main questions while we were doing this? So first of all, okay, of course, accelerated aging happens in Down syndrome. This is something that has been established. But we wanted to see to which extent does it happen. So how much older these people are than their peers, which is something that is quite tricky to determine. And also, when does it begin? But I've kind of spoiled that with the title, but pretend you didn't see that and then get surprised at the results later. And then we have another question is what causes these changes? So of course, it's the presence of an extra chromosome because that's what Down syndrome is. But you have kind of two groups within people who research Down syndrome. And this one group claims that it is this thing that they call amplified developmental instability that is actually triggered by a freely segregating extra chromosome. So irrespective of which chromosome, they are claiming if you have an additional chromosome, it is causing this chaos in the cells, and then you have this amplified instability as people age. But then you also have this other group who say, yes, it really matters that this is chromosome 21 because they claim that it's basically the consequence of a specific gene overdose. So one gene, or most probably, more genes for chromosome 21, whose extra presence is really making this happen. So you have these two groups, and we wanted to see, of course, we can't make the conclusion, what causes accelerated aging in general in Down syndrome, but we can say, based on IgG glycans, this is what we have observed. So these are our main two questions, and of course, to test them, we first needed some samples. So we had samples from 3 different European populations, from France, from the UK, and from Italy, and this is what they will be called later in the figures, just so that you get familiarized with that. And we had 246 samples of people with Down syndrome. 208 of them were adults, and 38 of them were children. Oops, sorry. And we did UPLC analysis of IgG glycans to quantify them. Okay. And when you do UPLC analysis, you get something like this. Maybe a lot of you have already been familiarized with this, but anyway, this is an IgG glycan profile. And over here, it has been neatly divided into different derived traits that we normally look at. So we look at G0, which are glycans with no galactoses. Then we look at G1, which are glycans with 1 galactose. Then G2, which are glycans with 2 galactoses. And G0 and G2 are the ones that I've mentioned that change with age. So this first part of the blue part of the chromatogram actually goes up as people age, and G2 goes down as people age. And then we have glycans with sialic acids, which are kind of in the later part of the chromatogram. These are, of course, not the only derived traits that we calculate. So we also calculate glycans that contain fucose, and we calculate glycans that contain bisecting glucose. We can also do a lot of different derived traits, but just for this research, we did these specific things. So to keep in mind what this graph actually means, you can here say that, you can see that over here we have, on the top of the panels, you have the names of the derived traits. So you have all of these are galactose related. So these are the ones with no galactose, 1 galactose and 2 galactoses. Over here are sialic acids, fucosylated glycans, and bisecting glucnak. And here in this orange color, that's the control population. And in the blue, that's the population of people with Down syndrome. And the first column is always France, then the second one is Italy, and the third one is the UK. So, of course, each of the control population was age and sex matched and demography matched as well. And over here you can see just when you take the derived traits from all of these populations and put them together, you can already, if you're looking at these asterisks, if you can see them, most of them are basically significant, so only bisecting glucnak isn't. But I just want to focus, just for the sake of time, on G0 and G2, which you can see that is higher in people with Down syndrome. For G0, and it is lower in people with Down syndrome for G2, which basically agrees with the fact that these populations are on average older than their age-matched and sex-matched populations. But what we wanted to also see, so over here you have kind of the entire picture, but we wanted to see how these glycans actually change with age. So then we plotted them against age, and over here you have a similar sort of a story. So you have France, Italy, UK. These are the galactosylated ones with no galactose. Then you have 1 galactose, 2 galactoses, and so on. And in these curves, you can see how the percentage of these different glycans changes with age. So down here we have age in years. And again, for the sake of time, I would like us to focus just on these traits that I have already mentioned, that they change with age. And how do we get to this number that people with Down syndrome are on average 19 years older than their healthy peers? So basically, if you look at the slopes of these curves, they are the same for most of them. They're quite similar. And they change in the same direction. So here in blue, you have people with Down syndrome again, and in this orange, you have the controls. So you can say that they're following the same trend at a similar speed, but the thing is that these curves are very obviously, vertically separated from each other. So what does that mean? Well, to demonstrate it, over here in G2, if you would look at the France population, and you're looking at the controls, so G2 drops as people age. And when it drops to around 15%, people who are from a regular healthy population, they will be 60 years old, right? But then by the time that they have dropped to 15%, people who have Down syndrome are actually just slightly above 40 years old, so not even 45. So that kind of demonstrates the point, you know. We have calculated this using these curves. And we have established that on average, when you take all of these populations into account, people with Down syndrome, of course, based on their IgG glycans, are 19 years older than their healthy peers. Then we have also samples of children, as I have already previously mentioned. So what did we do with the samples of children? Well, of course, we had a lot less samples from children, and we had this issue with finding controls, of course, because of the ethics of obtaining samples from healthy babies. We only had this control group who are all 4 years old, and they are all from Croatia. And then we had this group of children with Down syndrome who were also all 4 years old. They were from the UK. But we also have this here group, just for the sake of completeness, that we wanted to add to this graph, though the statistics was only done on between these two. But these children range in age from 1 to 5 years old. So these are all of the samples of children with Down syndrome that we had that we just wanted to show here. So if you were comparing then these two groups, you can tell that even when they're in this huge age range or larger age range, they're also kind of grouping with these children who are 4 years old. And you can see that they're basically following similar changes that happen to adults as well, specifically in G0. So this difference is significant. G2 is only nominally significant, but still it's following the same trend. So already in children who are as young as 4, you get a higher G0, and you get a lower G2 that also happens, is also observed in adults. And then an additional thing, I don't know if maybe somebody has noticed this little X on the graph. So that is the partial or segmental trisomy 21 part of the story. This child has been called Cro1. Why? Well, because it's from Croatia, but also it stands for critical region only, because this child does not have 3 copies of chromosome 21, but it displays a lot of the clinical symptoms of Down syndrome. So then when they looked into it, they saw that this child had a triplication on one of the copies of chromosome 21, so it had only 31 genes present in 3 copies, so no freely segregating extra chromosome or anything. And then when you look at it, so when we put it on the graph, here for G0, it is grouped with children with Down syndrome, so it's even outside of the range of the controls. For G2, it's, you know, it's below the median, but it is here in the range. So then we also did PCA to see where it groups, and no matter which way we did the PCAs, it would always group with the children with Down syndrome, which of course it is n equals 1, but it's kind of proof of principle that already as a child, with only these 31 critical genes, it's already showing these signs of you know, accelerated aging based on IgG glycans. So our main conclusions from this research would be that, first of all, IgG glycan measured accelerated aging is obviously very significantly pronounced in Down syndrome, and it also begins early in the childhood. And also that overdose of specific chromosome 21 genes is sufficient to drive IgG glycosylation change already. So these are kind of the conclusions from this paper, and this is something that's, you know, a finished story already published. But then, because we have been talking so much about IgG glycans, and I have been doing some more things that are not strictly IgG glycans, I just really wanted to briefly mention this collaboration that we had with Queen Mary University of London with Professor Cindrić group. And they have developed this really cool thing where they have an isogenic induced pluripotent stem cell model of trisomy 21. So what does that mean? So induced pluripotent stem cells, or iPSCs for short, they're actually cells. So you, for example, pluck a hair from somebody, then you grow fibroblasts, and then you reprogram them back into stem cells. And the reason why they're called pluripotent is because they can differentiate into almost any cell type that you would like to get. And why are they isogenic? Well, because they had an adult with mosaic Down syndrome, which means that some of this person's cells had 2 copies of chromosome 21, and some of their cells had 3 copies of chromosome 21. But otherwise, they had the same genome, of course, because they're, you know, from the same person. And they managed to establish this isogenic model. So these we call D21 because they only have 2 copies, so they are disomic for chromosome 21. And these we call T21 because they are trisomic for chromosome 21. And we didn't only have induced pluripotent stem cells, we also had neural stem cells that were kind of starting to differentiate into a neuronal pathway. And we did cell lysate glycosylation analysis, again using UPLC, but then after that we had to annotate the glycomes using tandem MS. And what we got here are the 2 chromatograms. They are just representative of all of the chromatograms that I obtained and annotated. And over here you can see that they're quite similar between iPSCs and NSCs, which we did expect. So they have these kind of 4 or 5, if you look at this one as well, major peaks, and they're all high mannose structures, which you expect when you are analyzing stem cells. Normally you get these more simple, evolutionarily older glycans and a bit less complex glycans. So this is kind of a well-established known fact. And you can see that I haven't separated them into, 4 panels that are disomic, trisomic, because we didn't get that many qualitative differences between disomic and trisomic cells. So if you look at just what each peak contains, there are some differences between iPSCs and NSCs, which is to be expected because they're different cell types. But then, for example, only here in GP17, we got a different structure for a disomic and trisomic iPSCs. So this here structure is dominant in this peak in trisomic cell lines, or trisomic for chromosome 21, and this here high mannose structure is predominant in the disomic cell line. So I found that quite interesting because that was the only qualitative difference between disomy and trisomy that I managed to detect. But then it's kind of hard to comment on just individual glycans. So what I did was I calculated derived traits here, but I didn't calculate the same derived traits as we do for IgG because that wouldn't really make sense. I wanted here to just separate them into the main glycan types. So you have these mannosidic glycans, then you have hybrid glycans, and then you have complex glycans. But then I also noticed that I had a certain percentage of these glycans, which I had kind of troubles with placing them anywhere because they're not really, according to Professor Varki's definition, they're not complex glycans, though some people call these glycans monoantennary complex glycans. Some people call them pseudo-hybrid, some people call them truncated glycans, but they definitely kind of belong into their own group, so I put them, as a 4th group here. And over here, I just wanted to show you, so I don't have any statistics for this yet. This is just a very, preliminary thing. But over here you have iPSCs, here you have NSCs, and then over here are the disomic cell lines, and here you have the trisomic cell lines. So when I kind of plotted these traits, and I put the numbers here just so that you would see it better. So if you would compare cell types, of course you get more or less high mannose glycans here in NSCs, you get more complex glycans, and you get more hybrid glycans, which you expect because as cells differentiate, their glycome becomes more complex. And then when you compare disomy and trisomy in NSCs, you don't really get, if you look at the numbers, if you look at the graphs, you don't really get that much of a difference. But then when you look into iPSCs, something interesting is happening, which I still don't know if it's really statistically significant, but each of the clones was acting in the same way. So yeah, we'll see. But then you get less high mannose glycans in trisomy, and you get a bit more complex glycans, and you also get more of these pseudo-hybrid glycans. So this also might be the case of, the cells kind of maturing on a level that we can't see by eye when you look at the cells, but maybe inside of the cell some things are happening earlier in trisomy than they are in disomy 21, which is, all still just very preliminary, and hopefully some next time I can show you when it's shaped into a paper and published as a preprint somewhere. But yeah, I just thought this would be an interesting thing to share with everyone. And that's it. I would like to thank our collaborators and also, of course, our funders for this opportunity.